I'm Masafuji, the leader of the WRHI Hub Satellite Lab. I'd like to introduce my international collaborators in this satellite lab. The first person that I'd like to introduce is the Professor Jim Rizzi from the University of Illinois. He's an expert of the cluster spectroscopies. Cluster means the molecular aggregate, so that's important the complex that we understand for the molecular recognitions. And the biomolecules often had a charge, like a positive and negative charge, so that means ions. So the Professor Otto Doffer is from the TU Berlin, is expert of the ion spectroscopies. And Professor Anzenacker is the expert of the chirality, and she's from the University of Paris-Saclay. And the chiralities means for the difference of the right-hand side and the left-hand side, and so this is the very important characters of the biomolecules. That's the important person. Also, she's an authority of this molecular understanding concerning the chiralities. And the molecules often has a photochemistry. After absorbing the photons, the biomolecules start to react. One of the famous examples is the photosynthesis in plants, but of course for our skin have some reactions, for example like sunburn, whatever. So we have the expert of the photochemistry in these teams. The one person who is the very famous and important person, Matania de Vries from the UC Santa Barbara. He has a new theory to the relation of the photochemistry to the evolutions. Another person is a younger generation who is an expert with photochemistry from the University of Paris-Saclay, Jill Gregory's. And we also need the help of the theoreticians to understand for what we measured. And we have the important two persons. One person is from the PNLL, the National Laboratory in the United States, the DOE, and the Professor Soteris Sanchez. He's an expert of the quantum chemistry and also they can control for the big computer facility in the United States. And another important person for the theoretician in the theory group is Professor Akio Kitao, who is an expert of the molecular dynamics simulations. And of course, for this team, we need something, the material, the person to can provide and handle the materials. The sub-leader of this team is the Michio Yoshizawa, who is an expert with the supramolecules. He can handle the very fine new molecular capsules, and that can recognize molecules. Also, for the biomolecules, the professor Tsutsumi is an expert of the peptide chemistry. He can provide for the knowledge and the materials of the peptides. In addition to these people, of course, for the inside of Tokyo Tech, we have the excellent people for the IR spectroscopy, U-based spectroscopy, like the Professor Shuichi Shuchi, the Professor Hirata, Professor Yasuhiro Oshima from the School of Science. And these people, they help me and help collaborate together, and they try to understand the basic understanding, the mechanism of the molecular recognition in such kind of living systems. The name of our satellite lab is Molecular Spectroscopy in Living Systems. So we are studying the molecular recognition in the international teams. Molecular recognition takes a very important role in the everywhere, particularly in the living systems. For example, in here, I'm showing the neurotransmissions. For example, like maybe you know that in neurotransmission, the neurotransmitter, like adrenalines, that they emit and accepted by the receptor in the synapse, and then all the neural system is working. And this receptor recognizes molecules, distinguish molecules. So that is called the relation is likely to the key and log. But it's something wrong for me because I show this molecular structure of adrenaline. That's they called in epinephrine in the US, and you can see there are so many single bonds. Single bond means for this molecule can easily change the shape can understand for the key that can change the shape, that soft key, that I don't understand why that's working. And also the receptor is the protein. Protein is not steel. That can change the shape. How can they recognize each other? That's my naive but big question. So to understand, of course, this is very important topic. So the people are focusing on it to understand for the how to recognize for the molecules. And probably one of the famous results, the great work is the X-ray diffraction work to obtain the beta-2 adrenaline receptor that bound the adrenaline itself. And so the people can know how the adrenaline is trapped inside of the receptors. That's great work. That's very difficult. Indeed, that's the Nobel Prize. So it's you can understand how that's important, how that's the innovative things. But even for this situation, people cannot understand why adrenaline can accept it, but the dopamine cannot be accepted. For this receptor, the dopamine, noradrenaline cannot drive. But you can see the structure is quite similar. Why? So we thought the traditional way may not be perfect to understand for the function of such a molecular recognition. So we thought about for the completely different way of the ideas. The molecular recognition takes place in a very local place, very little place. For this case, this is shown like SIDSF. That's the small part. That's the name of the sequence of the proteins. For this part, that recognizes the benzene part of the neurotransmitters. So we thought, okay, that happened local. Then we cut out for the very short part, and they try to make the complex that's bound to the other various molecules, like including for the adrenaline that can drive the receptor, the dopamine that cannot drive the receptor. Then they try to see how that is good, how that's wrong. That's our ideas. And to do that, we need a very advanced laser spectroscopies. And this is also make the very good base to understand how to think about how to design the drugs, how to design the new materials like nanomaterials for the innovative materials. Thank you for your attention.